this video, we're going to talk about translation. In other videos, we started talking about protein synthesis and the central dogma of molecular genetics. So transcription is going from DNA to making mRNA. And then translation is taking that mRNA, leaving the nucleus, going out to the ribosomes, and making a protein. And then to finish off the central dogma of molecular genetics, those proteins will go together to make your traits. Traits such as your hair color, your eye color, or how tall you are, etc. So in other words, in translation, we're going from a nucleic acid language in RNA and going into an amino acid language. And those amino acids will come together and bond together to make our proteins. When we go there, translation, we need to make sure that we have a few things sorted out before we start talking about things. Ultimately, like we said before, translation is taking an mRNA strand, and remember, this is a mature mRNA strand that only has exons. Okay, all the introns have been spliced out by the spliceosomes. So translation takes an mRNA strand, a mature mRNA strand, and makes an amino acid chain. So those chains of amino acids that are connected together, that is what a protein is, a chain of amino acids. All of this process of translation happens at the ribosomes. The ribosomes are located in the cytoplasm of a cell. So if we have our cell, and here's the nucleus of the cell with the DNA on the inside, all of this space in between the plasma membrane on the outside and the nucleus on the inside, all of this space in here, that's all cytoplasm. So we might have a ribosome here and here and here. There's a bunch of them all throughout the cytoplasm. There's four main things that are involved in translation. Obviously, our mature mRNA, tRNA, which we'll talk a little bit more about in a minute, amino acids that come together in a chain to form our proteins, and then it all takes place on the ribosomes. And this is kind of a big picture of all of protein synthesis. So we start with our DNA that happens in our nucleus. The DNA strands get pulled apart, and the DNA template strand, we make our mRNA. Then our mRNA is going to leave the nucleus and go out to our ribosomes. And then from our mRNA on our ribosomes, we'll make our chain of amino acids, or in other words, make our protein. I know we've had a lot of vocab, but we got to take care of a little bit more vocab. A 3-nucleotide sequence of mRNA. Those are called codons. Codons are going to be very, very important, as we'll see in our next slide. There's not very many codons that you have to memorize, and in fact there's only one, and that's AUG. AUG is the start codon. All translation starts at the start codon. So we can have a bunch of amino acids, or nucleotides, we can have a bunch of nucleotides here, but until we see that magical AUG, nothing else before it matters. Nothing matters before it until we see the start codon, which is AUG. And then it'll continue on until we see a stop codon, but that'll become more clear in the next slide. We don't have to memorize that. We just have to memorize AUG is the start codon. Each codon codes for one specific amino acid. And we'll see again in the next slide that the codon UUU codes for the amino acid phenylalanine. Last bit of vocab. A 3-nucleotide sequence of tRNA is an anti-codon. Keep the T's together. tRNA is an anti-codon. So 3-nucleotide sequence of tRNA is anti-codon. A 3-nucleotide sequence of mRNA is a codon. So what are the codons? Here we have our codon chart. This is all of the code for all of the life. and it doesn't matter if we're talking about seaweed, a banana tree, a human, a chimpanzee, your pet dog. This is for all life. And we're talking about here specifically codons, which is mRNA. So 3 base pair sequences of mRNA. Here's the chart we're going to use. Remember before I said U, the codon codes for phenylalanine. Well, here in our first base, there's a U, second base, U, third base, U, codes for this amino acid, which is phenylalanine. What would, let's see, A, C, C code for? Well, the first base is A, C, C. There's the amino acid. The code is redundant. What we mean by this is if you look at, let's take this group right here. CUU, CUC, CUA, and CUG all code for the same amino acid, which is leucine. So a lot of times this third base, a lot of times it doesn't matter what it is because in this, as we see in this example, they all three code, or all four, excuse me, code for the same amino acid. Like we said before, the start codon is AUG, and that technically is methionine is the name of the amino acid that it starts with. And then there's 3 different stop codons. We see those over here. Okay, you do not have to memorize those in any way, shape, or form. We just need to know that they exist and what they are and be able to use them. Notice that there is no amino acid for a stop. Stop just means stop. Stop is not an amino acid. How are the codons matched to the amino acids? Well, remember that we start with a DNA molecule. And let's say that this top strand is the DNA template strand. So we need to have the process of transcription happen first. So RNA polymerase is going to break the hydrogen bonds and separate those 2 strands of DNA. But for right now, let's just focus on the DNA template strand. Transcription happens, so we make our mRNA strand. So as we see here, our U's bond to and match up with an A, C's with the G's, T on the DNA will make an A on the RNA, so on and so forth till the end. Now, remember this mRNA strand is going to leave the nucleus and go out to the ribosome. Once it's out in the ribosome, okay, so now this DNA is gone, right? It's not in the same spot. That DNA is now in the nucleus. Okay, it's not at the ribosome. Okay, now this mRNA is out at the ribosome, and what we're going to have is we're going to have our tRNA bring over our amino acids. So our tRNA is directly connected to an amino acid. Remember, the 3 base pair sequence of tRNA is called an anticodon. And we can't start before we start, so it means that we can't start until we get an AUG, which we have right here. So what should match up with an AUG if we're talking about RNA? Because remember, this is tRNA. Well, we have UAC that's connected directly to this AUG here. It's connected directly to the amino acid. This anticodon will bring this specific amino acid here. GCA will bring in this specific amino acid to the ribosome. GUA would match up with CAU and bring over this specific amino acid. Here's just another quick diagram about that as well. So maybe we have a few A's, U's, A, G, etc. before that, but remember, none of this matters until we get the start codon of AUG. Then we'd have another tRNA anticodon and attached to its specific amino acid waiting to come over and for the ribosome to grab it. So after the start codon, the ribosome will take the amino acid that's right there and grow that amino acid into a big long chain like this. So the tRNA, after the amino acid has been taken, that tRNA leaves, the anticodon leaves. The ribosome will move down through the next chain, grab the next amino acid, attach it to the chain, tRNA leaves. The next tRNA will come down into the chain, match up perfectly, right? UUC, AAG, connected to its specific amino acid, will come in and attach, waiting for that amino acid to be grabbed from the ribosome. And what we're left with is a big, long chain of amino acids. All of this will continue in a similar fashion. New tRNA with amino acid attached come in, steal the amino acid, put it in the chain until we get a big long chain, until we get, it will all continue until we get the stop codon. Protein synthesis. Let's break down that word. Well, synthesis means to make. So therefore, protein synthesis means to make proteins. More specifically, protein synthesis is the process of transcription and translation both together. So the part of the central dogma that goes from DNA to RNA to proteins, all of that is protein synthesis. Here's the whole big picture of protein synthesis. We have our double-stranded DNA that's in the form of a double helix. Once we receive a signal to express a gene, RNA polymerase will break the hydrogen bonds and bring in free-floating nucleotides to make an mRNA strand. Remember, that process is called transcription. After transcription has happened, the mRNA strand will leave the nucleus and go out to the ribosome, where the ribosome will read in groups of 3 base pair sequences of the mRNA, which is called a codon. And we got to wait for the start codon, that is AUG. The tRNA, and more specifically the anticodons that are attached to specific amino acids, the tRNA anticodons will match up with the mRNA codons and bring over the specific amino acids and grow those amino acids in a chain until we get a stop codon. And now we have the proteins made. And that is all she wrote.
It's like this and like that and like this and it's like that and like this and like that and it's like this. So just chill till the next episode.